Hi everyone, my name is Kristine Roberts and I'm a PhD candidate at University of Aberdeen just now, and I am being part funded by MND Scotland. And I attended the MND Scotland LEARN event in Aberdeen, and we wanted to make this presentation available for everybody to see. So I'm at the start of my journey, but I thought I'd give you a bit of insight into what I hope to achieve through my PhD. So bit about where I came from. So my experience, I trained as a biomedical scientist in the NHS and specialised in pathology. And then I moved into a role in a tissue bank. And my main role here was to get tissue samples into research. And it's this kind of experience that I think MND Scotland kind of recognised that is needed to move MND research forward. And the other experience I'm bringing into this is my master's in stratified medicine, which is about personalising treatment. I'm going to explain all these terms as I go through, and using biomarkers, and the whole kind of idea of stratified medicine is getting the right treatment for the right person at the right time. And I hope to bring all this experience and apply it to my PhD in motor neuron disease. So a bit about my research interest coming into this. So biomarkers of disease. Again, I'm going to explain these terms just to give you a bit of background here, and about how we use these biomarkers to use them to make diagnostic tests. And we use these biomarkers to group patients based on how the disease is behaving, and then we can personalise medicines. So we can give patients treatments that we know will work for them based on these biomarkers. I'm also interested in the immune system and how this works, and in disease, and especially a particular group of cells called T cells, and also other markers of inflammation. And we want to see if we can manipulate the immune system in disease and intercept it with drugs to try and slow or prevent disease. So these are the kind of things I want to bring into my PhD in motor neuron disease and hopefully further research, with the intention of getting an early diagnosis and targeted treatments for patients. So first of all, what is a biomarker? So you can see it comes from biological marker, that's how it's made up. And biomarkers are a snapshot of what's happening in the body at that certain time. So technically weight, blood pressure, these count as biomarkers. But what we're more interested in are biomarkers on the protein and molecular level. So DNA, electrolytes, that kind of thing. And just for some examples, you've maybe heard of prostate-specific antigen, or PSA, and this is a blood test that males can get to see if they have an increased risk of prostate disease, and also the bowel screening program. So these tests rely on biomarkers to inform us if patients are at a higher risk of disease. We need biomarkers to be accurate and reliable, so a lot of work goes into making sure that we can trust them, basically. And especially if we're going to give people treatments based on these biomarkers, we need to know that we can trust them. We also need to know what the biomarker means in relation to disease. So how high is too high? So we have these ranges of normal, high and low, but it takes a lot of work to establish what the normal range is. So we need to understand this in relation to the disease. And we also use them to see how people will respond to drugs. So a good example is insulin. So diabetics use their blood sugar. That's kind of a biomarker of how their body is responding to insulin. So how can we use biomarkers effectively? What we need to do is we need to make these biomarkers into a diagnostic test. And this is what we hope to do in MND. It's kind of the focus of my project. And what we're particularly looking at is samples which are easy to access. So not the brain, not the spinal cord, which is where you typically see signs of disease, but actually in gut and skin biopsies, we've seen some signs of disease. So this is a kind of diagram to explain what we have been looking at in the lab. So the picture on the right, the microscopic picture, is some staining that I have done in the lab. And just to explain it, so if you have the whole large bowel on the left-hand side, and in the middle, this is if you took a little piece out of the wall of the bowel, and you can see on the top, they've got all these little finger-like projections, we call them, called villi. And these are there to increase the surface area of the bowel. And it just increases absorption and increases blood flow, things like that. And then, so when you look at this microscopic picture on the right, these little folds that you see, or crypts we call them, are these villi. And so the surface is on the right-hand side here, and this is kind of all open to the inside of the bowel. And I've tried to put little arrows here. It's maybe a bit tricky to see here, but the really dark brown spots are the certain cells which are staining really positive with this marker. And it's an investigative marker at the minute, but if we're seeing that this marker is increased in MND, there is potential that because these cells are sitting in the bowel, could they be coming away from the lining of the bowel and going into stool samples, which gives us an opportunity for an easy diagnostic test. So this is something we're looking into, and we're hoping that we can maybe make it into a point of care test, so a bit like bowel screening, or like the dreaded COVID test we all know how to use. But as you're aware, these are really simple tests, and they could even be done at GP practices as early markers of disease. And if we can't see it in the stool samples, even adding on an extra test to colonoscopy, endoscopy biopsies that patients routinely get if they're having bowel issues, it's an easy thing to do. It doesn't mean an extra biopsy, but we can add a test on in the lab. So these are options we're exploring. We're also looking at skin. Obviously, it's easy to access as well. So next I want to speak about how we can use these tests to treat patients effectively. So very good, we've got this test, but what next? So this is where I want to introduce the concept of personalised medicine. So to illustrate this, if we have the whole MND population sitting over here, but we've developed lots of tests for lots of different biomarkers that we hope to find, by doing all our work in the lab and clinical trials, we can hopefully work out what drug will work for each person based on those biomarker tests. So this blue group might test for positive for one biomarker, which we then know one drug will work for these people, but it won't work for the green people. We've got another drug for them. So this is what we hope to achieve, and this is something that's routinely done, particularly in cancer. So an example with breast cancers, if you've got a whole group of people with breast cancer, we test for estrogen, progesterone, and HER2 markers. And depending on the results of these 3 tests, patients are given personalised treatments that we know will work. So this is what we're hoping to get to in MND. So bringing it back to MND and the immune system, which is where I'm looking for these biomarkers, where do we start? So this is just a snapshot. I know you can't see it very clearly, but it's just a snapshot of one pathway in the immune system, and there's so many proteins, so many cells involved in this process that it's a bit of a minefield. So where do we start? So I'm first of all focusing on a group of cells called T cells. So what are these cells? T cells, they come from the bone marrow, but they mature in the thymus, which is a gland that just sits in your chest here. And they make up the white blood cells in blood, basically. So they migrate around the whole body, and when they're doing their job, what they're meant to do is get rid of anything nasty, so infection or unhealthy cells, and technically they should get rid of tumour cells when they're working properly, and it's when they start to not work properly that they can't get rid of the tumour cells. So what we're seeing is that these cells are altered in MND patients, but not every patient is showing the same change. So this is what we hope to look into a bit more. And again, how do we know where to start? Because T cells, there's still lots of different types of T cells and lots of proteins involved in these processes. So the first thing I did was I did a literature review of everything we know basically so far. So I looked at every single study that's been published looking at T cells and the immune system in motor neuron disease. And there were 3 main findings from this. The first one was that the immune response is different between patients. So we're not seeing everyone's immune system behaving the same, which is interesting and offers up the opportunity for personalised medicine approach. I also came across multiple papers that saw a split in how patients' immune systems behave on a cellular or genetic level. So patients would maybe split into 2 distinct groups based on these different immune markers, but they weren't always split based on the same measures. So one paper said that a specific type of T cells called Tregs, if patients grouped based on low numbers of Tregs or high numbers of Tregs, and another study found that patients grouped based on rapid versus slow disease progression, and another paper was with or without cognitive decline. So there's no consensus really on what this split is and what's causing it and how we can measure it in the clinic, kind of thing. And interestingly, once I looked at all the numbers, 2 markers were consistently elevated across studies. So these 2 were interleukin 17A and interferon gamma. So these are 2 that we want to look into a bit more and see if they can be targeted to reduce them and see if that can improve disease. So I'm talking about immune therapies, and this is not a new concept. Immune therapies are used in lots of other diseases, autoimmune diseases and cancers. So this is what we're looking to try and mirror what's already done, because there's loads of drugs out there already, and could we just repurpose these to target the immune system in MND? So just to explain how T-cell immune therapeutics work. So we've got a wee example here. For example, in melanoma, T cells are reduced, and that's why the cancer is being allowed to grow. But in rheumatoid arthritis, T cells are increased, and this is why you get increased inflammation, and that's what is causing the disease. But interestingly, both these mechanisms are caused by the same marker, SCTLA4 it's called. And basically what we want to do is re-establish this balance of the T cells in the bottom here. And there's 2 different drugs available. So ipilimumab in melanoma will increase the T cells, and that will help to get rid of any remaining melanoma cells. And abatacept in rheumatoid arthritis will reduce the T cells and reduce inflammation and give patients a bit of more comfort from the inflammation. So potential for the future, the aim of the PhD really, and our further research and our research as a wider group, is to identify reliable biomarkers that we can implement in diagnostic tests and hopefully achieve earlier diagnosis so that patients can get support earlier, and we can hopefully personalise therapeutics based on these as well, with the ultimate goal, of course, with slowing or preventing disease. So thank you very much for listening. I'm really happy to take any questions if anyone wants to get in touch. My email address is in the bottom here. And just a massive thank you to MND Scotland and the Chief Scientist Office for funding this work. This is the rest of my team up here. Of course, thank you to those with lived experience of MND for taking part in research, because without you guys, we absolutely could not do our work. Thank you.